Dear learners, welcome you again in the course of Biodiversity Conservation. Earlier, we have discussed about the field and herbarium techniques. Today, we will continue the herbarium techniques and we will explore the some key techniques in herbarium specimen preparation. As earlier, we have discussed about the collection of plants, and today we will focus upon the after the collection, the next process, that is the pressing, drying and other techniques that use in the preparation of the herbarium. So, these techniques are essential in preserving the plant specimens for long-term study. So, let us go through the steps to create an effective and durable herbarium specimens. So, these are the 8 main steps in creating the herbarium specimen, like the collection. This is the first one which we have discussed earlier. Then today we will going to discuss about the pressing, drying, poisoning, mounting, stitching, labeling and storage. These are the 7 next steps after the collection for the preparation of a durable and plant herbarium. So, each step plays a vital role in preserving the structure and visual integrity of the plant specimen. So, today we will focus on the pressing, drying and the other steps. So, let us discuss the first next step in the plant specimen preparation, that is the pressing. So, first one as mentioned here, that is the pressing. The pressing is a critical step that requires enough pressure to flatten the plant while preserving in botanical details. And pressing allows for a neat display of leaves, flowers and other structure that can be studied later. So, using a plant presser and appropriate paper is essential in this particular process. As you can see in this particular image, this has the plant press. That is a very important in the procedure of the pressing. So, here this is the list of the material that required for the pressing, as we use a specific material to press the plants, which include a plant press as we have seen in this particular image. Then the next one is the newspaper, because during the pressing we have to press the plant in between the newspapers that absorb the moisture. Then greaseproof paper in case of a aquatic or very wet plant materials, then blotting paper for the removal of excess moisture, then corrugated cardboards for the proper ventilation of the plant material. And each material has a particular purpose, like newspaper absorb the moisture, blotting paper speeds up the drying process and cardboard aids the ventilation as I have told you and preventing mold. So, these are the some required material that required for the pressing of the plants. Then the next you can see here, these are the cupboard. You can see here in this particular image, these are the cupboard which is for the plant press material. Then these are the plant in between the newspapers, you can see here, which absorbing the moisture. These are the some material that required for the pressing of the plant. Then the next step is the procedure of the pressing. So, before pressing, ensure that the specimen is clean and centered on the pressing sheet. As you can see here, we have to place the specimen in the center, and if the specimens are larger enough, they should be bent, giving them in the shape of a V, N or W. So, depending upon the size of the specimen, we have to bend it like in the shape of V, that reduces the size into the half, then N, that become the specimen become the 1/3, and in case of W shape, the specimen become the 1/4 of its actual size. So, the bundle should be uniform in thickness in the middle and on the sides, and the specimen should be kept on one of the above other. Then the next in the pressing procedure is the plant press is used to press the plant specimen, which is very important, and the press only one specimen per page, because if we mix the specimen, that will cause the intermixing of that particular plant parts. Then the leaves, flower and other structure should be arranged in as natural as manner as possible on the newspapers, while at the same time trying to avoid the excessive overlap and crowding. And trim the excess material in a way that is clearly observed that the material was removed from that particular material from that particular place. Then Arrange the leaves, flower and other parts as naturally as possible to preserve the overlap. Be sure to show both surfaces of the leaves like dorsal and ventral to display specific characteristic, and the reproductive structure should also be spread open for easier examination, and at least some flower should be spread open to show the internal structure will be visible for the examination. When you have finished the arranging the specimen with the newspaper sheet or combination of a paper you have chosen, then we have to need to intersperse them between the corrugated cord sheets to aid the ventilation, and finally we have to place everything in our press and tighten well, and change the paper after the 20 hour till it completely dried. So we have to change the papers on every 20 hours, and 5 to 10 days is typically for plants in a press without any heat source to get dried. This is the simple procedure of the pressing of a plant. And after the arranging, place in between the plant press with sheets of cardboard between each layer for ventilation. Then we have to tighten the press to apply the consistent pressure, which helps to remove the moisture. As you can see here, this process should be checked regularly for the first few days to ensure the proper drying. So as we can see here, the first 2 days or 4 days you will need to check daily and change the blotting papers and other surrounding papers, and the retighten the press, but as the plant dries, the check and can become the less frequent. As you can see in this particular image, this is the wrong method of pressing the plant, because you can see there is no proper shapes of leaves is not visible, no flower is, the shape of the flower is not showing in this particular image. And this one is the right method of pressing a plant. You can see the arrangement of leaves is clearly showing, and the leaves which is a, you can see this is a leaves characteristic is clearly visible in this particular, and the flower, each flower part is quite visible in this particular pressing plant material. And when this type of a, when we have spreaded the plant parts, as you can see here, we have to keep it in between the newspapers or blotting papers. Then as you can see in this particular image, we have to place them in between the plant press or cupboards, and then we have to tighten that particular, as you can see in this particular image. This is the procedure of the pressing of the plant. And this particular figure showing the sequence in which plant press is packed. As you can see here, these are the strap with D-rings that for the tightening of the plant press. Then this one is the wooded lattice frame. These are the cardboard that is important for the pressing of the plant. Then here you can see the 2 sheets of drying paper. Then this is the film sheet with specimen, as you can see here. Then another, there is a 2 sheets of a drying paper. Then further there is a film sheet with the specimen. Then further there is a presence of a 2 sheets of a drying paper. So you can see these are the specimen sheets. Then it here upper and lower, both of the side they have a drying sheets. Then they have a cardboard and the wooden lattice frame. Then we have a strap. So we have to tighten after the placing the specimen. We have to tighten this particular strap that for the important for the pressing of the plant material. So this is the steps. All the steps are and images at the material required for the pressing is shown in this particular image. Now after the pressing, the next step is the drying. As you can see, when we have pressed our plant properly, then we have to dry it. And after pressing, the next step is drying, as I have told you, and there are 2 primary drying methods: natural drying and artificial drying. So the first, that is the natural drying, is the after the pressing, the next step is the drying. So after the pressing, we have to, after 2 to 3 changes, the specimen are finally kept in a fresh blotting sheets and then dried by either natural and artificial heating. So these are the 2 methods of the drying. The first one is the natural drying and the next one is the artificial drying. And in case of a natural drying, the pressed specimens are left in the press for 1 to 2 days before being transferred to the new blotters. And in the course of 48 hours, the plant must be changed at least 3 times. This will hasten the drying process and prevent the withering of the plant parts. Then the sheets must be completely dry and free from mold, and if sheets found to be infected, they must be discarded immediately. And after for 1 week, there must be a daily changes of the sheet in case of a natural drying procedure. And for the artificial drying, which is a more faster procedure, requires approximately 12 to 48 hours, and a drying press with controlled heat source like kerosene lamp or low temperature oven can be expedite the process. So the heat should be kept below 50 degrees centigrade to avoid the damaging the plant tissue. As you can see, this is the artificial drying process with the kerosene lamp, and this one is the older form. As previously in the image we can see, the oven is now quite new method that is used for the drying of the specimens. After the drying, the next step is the poisoning. Poisoning prevents the decomposition and insect damage by killing the plant tissue, thus stopping the abscission. So, traditional poisoning method uses mercury chloride in ethanol, though safer methods like formalin and ethyl alcohol are now common. So, poisoning is done after the drying and before mounting. As you can see here, these are the some methods. Like the older method is with the mercury chloride that prepared in a ethyl alcohol, and for this process we have to dip the plant in this particular solution, and for this we have to use the dipping the plant in this particular solution with the help of some forceps, and for then after the pouring the specimen, and then we have to remove from this particular solution. So, dipping finger in the solution should be avoided because as we know the mercury chloride is a quite poisonous. So, we have to wear the gloves in our hands. Then the next one is the lauryl pentachloride. It used in some herbaria as a substitute for the in case of a mercury chloride. As I have told you, the recent method is the formalin method, has often been adopted for the collecting and poisoning a plant, and the collected and spread out in the ordinary old newspapers and bundled up. In this particular case, the bundle are placed in a polythene bags and soaked with the 70% ethyl alcohol and 10% formalin mixture. And the bags are tied airtight after the ensuring that the bundles are thoroughly soaked but without excess formalin. So, this is the method of a poisoning to prevent the plants from the drying and damaging from the insect. Then the next method after the pressing and drying or poisoning the herbarium specimen is the mounting. And as we have seen that after the specimen is pressed, dried and poisoned, now it is affixed along with the label on the mounting sheet. Or we have discussed earlier that the mounting sheets are made from the heavy long lasting white card sheet with the uniform size approximately 28 into 40 centimeter that is used in India, and the aim of mounting is that the specimen should be neatly and uniformly spread and fixed on the sheet, and all parts of the plant should be easily visible for the study. So, the common technique now in use our country is the pasting a specimen to sheet with glue, because glue paste is prepared by adding flakes of glue to boiling water and gradually and in small quantities till it makes a thin syrupy paste. So, this is the method of the mounting of a plant specimen. So, this give the glue some insect repellent property, and a small quantity of a mercury chloride, thymol crystal or copper sulfate, that is blue vitriol, is added in this particular glue, and the glue or paste is coated on a glass plate or tin plate, and then a specimen is placed over the paste and tapped gently with the forceps, and the mounting sheet with specimen glued on them are kept in the press for 1 day for proper sticking and drying. As you can see in this particular image, this scholar is mounting the specimens on the herbarium sheet, as you can see here, and this one is the mounted specimens on the herbarium sheet. And after the mounting, the next step is the stitching. Stitching adds further stability to the mounted specimens because small independent stitches are made on the each side of the stem or twig, because as you can see here, some plants may be not properly glued due to their small part portion. So, they may be get damaged during the transportation. So, this prevents the specimen from the shifting and ensure the durability, and especially for the larger or heavier part. As you can see, this scholar is stitching the specimen for increasing its durability. Then the next is the labeling. Labels are crucial for the documentation, providing details about the specimen's origin, habitat, date of collection and collector's information. So labels are typically placed on the bottom right of the sheet, allowing future researcher to easy references of the data. As we have earlier discussed that during the field trip, we have to prepare a notebook. That notebook is very important because now we have to enter that information in the herbarium sheet. So that notebook is very important during our collection because now we are going to write down all the information that we gathered during the collection on the labeling. So the labels are put on the right hand side at the bottom of the herbarium sheet. As you can see, this one is the image of a label, and they typically have a size of a 3 into 5 inches size and should have the following information, as you can see here, like name of the organization with a specimen from the plant originated, then name of the family of that particular plant, then botanical name of the plant, local name, locality of the collection, date of collection, habitat of the plant, field notes and collection number, and name of collector. So as you can see, there is so many information, like this one is the name of organization, that is the Central National Herbarium, that is Calcutta, that one is the India's largest herbaria. Then you can see here, that is the Botanical Survey of India, then the Flora of Darjeeling district, then this has a particular collection number, then family of, that is the Orchidaceae, then name, local use, then locality and notes which we have to write down during the collection, and the collector name, that is the H.J. Chaudhary, and identified by also by the H.J. Chaudhary. These are some information that we have to require and we have to paste in the form of a label during the herbarium sheet. So this is a typical example of a herbarium label. As you can see, like the scientific name of the family, like Ranunculaceae, that is the Ranunculaceae ficalia. Here you can see this is the scientific example of a herbarium label, like scientific name, that is the family Ranunculaceae, and the name of the species is Ranunculus ficaria, and the genus and which is the species. Then vernacular name of this particular plant is the Leocel calendrine or pilewort. Then the collector name and specimen name is the Lawrence. Then date of collection is the 20th March 2003. Then the locality is the Alden's House Gardens, Twickenham, England. Then habitat like damp clay soil and 20 yards from the river bank, growing in a damp shade on the edge of a deciduous woodland, and nearby plant in dock, that is the Rumex up to foliage. Then habit is the perennial herb up to 20 cm tall with stem creeping and rooting, and the characteristic the leaves hairless, glossy green, and flowers bright glossy yellow and turning white with age. So these are some characteristics of the plant that is the Ranunculaceae ficaria. As you can see here, these are some herbarium sheet. You can see these are the sheet, and this one is the label you can see here. So we have to place the label from the right corner of the herbarium sheet, as you can see here. And you can see here, depending upon the size, this one is the V bend because the plant size is larger, so we have bent it into the V shape. As you can see here, this also bent because the size of the plant specimen is larger. Then this is the typical characteristic of a good specimen. As you can see here, all the information that is gathered is placed in this particular specimen. You can see here, all the flower part is preserved. They have a specific label. They also have a seed and flower in the, you can see in the pockets. So it is providing all the information that is necessary for the identification of plant in the form of a herbarium. Then after the collection and pressing, drying, mounting, that we have created a herbarium specimen. So now when we putting a label, that we have identified that particular specimen. So for the identification, the scientific method is to first study the character of the plant and then check them with the flora of the region, work through the family and genus species, and key and compare with the full description and illustration. And thereafter, it is to be carefully compared with the earlier identified plants of the species or variety as the case may be. So, it is very important for the determining identification of the plants, because that we are submitting into the herbarium and that will be used for the future study. So, it is a very critical step. That's why we have to note down all the characters of that particular plant. Identification is a very important step as we have discussed earlier. So, it is a very important process because in future all our future generation will study from this particular herbaria. So, we have to write all the information very carefully in this particular herbarium sheet in the label. Then when the specimens are ready, that is mounted, labeled, identified, they are stamped with the distinctive mark of the herbarium or institution. And the stamping is usually done on the top right hand corner of the sheet, and this stamp carries the name of the institution, a serial number called the herbarium association number, and something the date of association. So, the mounted, identified and associated herbarium sheets are sorted out family, genus and species wise. So, after this process, now we have to store that particular specimen. So, plant specimen which have properly mounted and identified and filled systematically in the special wooden steel cabinets. So, the herbarium sheets loaded with specimens are filed inside a folder which are of various color schemes indicating the species, genus, family and geographical area. Then plants are arranged and stored following the either Bentham-Hooker system or England-Planter system of classification, which we have discussed earlier. In India, generally we follow the Bentham-Hooker system of classification in our herbariums. Like you can see here, this is the arrangement of a specimen. The specimens are usually arranged in the herbarium according to some recognized system of classification as we have discussed earlier. In many Indian herbaria, the order and the numbering of the families is generally according to the Bentham and Hooker. That is, you can say genus Plantarum. As you can see, this one is the herbarium cabinet. And in few herbarium, European countries, the herbaria of the families are arranged according to the recent classification like APJ. Then the herbarium sheets are to be arranged in wooden or steel almirah with pigeonhole compartments. As you can see here, this is the herbarium cabinets. You can see further, there is the images of herbarium almirah which has a pigeonholes. Further, you can see these are the almirahs with herbariums which showing the pigeonhole. These are the some boxes that have some characteristic of the particular flower parts or other parts which cannot be available to the press. So, we have to put them in a boxes. Then the next step is the fumigation. Fumigation is done for killing the insects which often damage the plants. So, the most common method is to keep naphthalene balls within the muslin bags in each pigeonhole, and sometimes parachlorobenzene is also used for the fumigation. And the small cloth bags with PDB are placed in the pigeonholes, and the chemical also be sprinkled on the sheets or bundles for the process of the fumigation to check the damage of the plants. Then the last one is what is the safety measure for sending a specimen. So, once the specimen has been dried, then are mounted on a paper sheet. Suppose we have to send the specimen from the different places. So, for this the plant specimen can be strapped with liner or cotton thread and knotted on the reverse side of the sheet or attached to the paper using thin strip of a transparent adhesive tapes or cellulose tapes. Then care should be taken while packaging to avoid the damage during transportation, and the specimen sheet should be put between the covers and shredded papers or thermocol sheet to fill the empty spaces. So, these are some safety measures that is important for the sending a specimens. So, today we have learned about the methods of preparing a herbarium from pressing and drying to the fumigation. This all increases the durability of the plants. So, previously we have discussed about the collection of a plant. Then today we have discussed other procedures for the preparing a durable herbarium. I hope you have enjoyed this lecture and learned how to prepare a herbarium, as we have discussed each and every step very clearly. Hope you enjoy this session. That's all for today. Thank you all. Thank you so much for joining this session.